Thank you. Thank you, Tony, for arranging this date. I will need to darken the room at some point. Let me just try. This is tot. How do we turn off these guys?
Right up here. This one?
Okay, that's good.
Do you want it like that now? Or?
Yeah, let's just leave it like this. So it's really a great pleasure to be here. Thank you, Raphael, and thank you, the students and Ariel, for arranging this. And it's a great to be back in Boulder. I've been here several times, but many years ago. I think last time was 6-7 years ago. It's always so beautiful here. So what I'll try to do today is make a young professor mistake and try to give you 2 talks in a period of 1 hour. And it's not going to work, so we'll see how we do this. My group has been working on quite a few things. More familiar topics, which are actually not on the list, are single molecule threat, a method that we introduced and now used by many groups, and the introduction of quantum dots to biology together with polarizators. More modern topics in the lab have to do with super resolution, voltage sensing, single cell physiology, development of novel detectors, and the combination of single molecule methods with optofluidics. I'm going to discuss today 2 topics if time allows. Most likely I'll cover maybe only 1 topic, but we'll see. So I already discussed during the day with several people here, and it seems like many people have heard about the SOPHY super resolution method that was originated. Really, the idea is not my idea, it's a Jörg Enderlein idea, but the work was developed in the lab. So that's what we are going to cover mostly, and if you have time, this is a recent topic. There is a paper that was just published. All right, very quickly, excitons in semiconductors. I'm talking mostly to double E, or there are some biologists here beyond Dylan and Yazan. Anybody else? Biology, chemistry, engineering, physics? Okay. All right, so this is probably familiar to most of you. Bulk semiconductor have this energy momentum dispersion relationship. This is a valence band, conduction band. We promote an electron from the valence to the conduction, and we generate an electron hole pair. And to all aspects, we can think about this electron hole pair as basically a hydrogen atom with a Bohr radius. This is the exciton, a quasi-particle. And the exciton has unique features in the absorption, in the density of states. If you cool a bulk semiconductor down to a very low temperature and you look at the absorption, you get the square root of E dispersion relationship plus a series of atomic-like transitions that are due to this excitonic feature. When you heat up the semiconductor to room temperature, some of those features will end up with a small peak called the exciton peak. And if you put such an exciton into a box, you basically take the original Bohr radius of the exciton, and now you want to squeeze it into a small particle. If the box size is larger than the Bohr radius, you don't expect to see any changes in properties, but if the box dimensions is comparable or smaller than the Bohr radius, you are in the quantum confinement regime. And this quantum confinement basically allows you to shift energy levels. So you take a particle in a box, and you change the box dimension, the energy spacing between states get larger. And this has been beautifully materialized in the early 80s in Bell Labs and IBM by developing new synthetic methods for layered material like MOCVD and MBE. So the era of quantum wells and engineered wave functions for electronic and optoelectronic devices was developed. Further, people developed one-dimensional wires, basically confinement along 2 dimensions, and eventually quantum dots where the confinement is in all dimensions. You expect to have vast changes in density of states and in absorption depending on the geometry and how many dimensions you confine. So in the mid-80s, both in the Soviet Union and in this country, people developed methods to make such quantum box with 0 dimensions, basically confinement in all 3 dimensions. Obviously, to make confinement in 1 dimension or 2 dimensions, there were established methods, as we discussed, MOCVD, MBE, and so on. To do this, that was much more difficult, and for that reason, people looked for chemical methods of making those little tiny semiconductors that are sphere-like box in all dimensions. The group of EFROS in Soviet Union, in Moscow University, and the group of Lou Bruce in Bell Labs developed those chemical methods of making colloidal particles. And we'll talk about synthesis in a second. Lou had really 2 talented postdocs at that time, Munjib Wendy, who moved to MIT and had a stellar career there, and Paula Vizatos, who moved to Berkeley and had a stellar career there. And I was lucky to interact and work with Paul. The synthesis is very simple. You basically take a flask, you put liquid soap in the flask, you heat up the soap to around 300 centigrade, and then you inject abruptly, just with a syringe, these metallo-organic precursors that contain the metal ions that you're interested in. The metallo-organic precursors basically separate into the individual ions, and you get nucleation and growth. And luckily, if you work out the conditions, you can get to the point where you get the growth of those particles. They start from a small nucleus and they grow up as a sphere, and they all do the same. This is really a very highly nonlinear process where you get very nice control over size and shape. And here you can see this is a high resolution TEM that shows atomic columns, showing basically how good the crystallinity of those materials is. And here is a monolayer, dense monolayer of these particles, almost arranged to a perfect hexagonal array, just showing that all those particles are of the same size. So you can make those particles, and I was lucky to, at that time, to come up with the idea that there are really good advantages for the optoelectronic properties of those particles compared to conventional organic dyes as it comes to biological imaging. And here we'll go through a list of advantages. The most important advantage is that you still have bulk properties for the absorption of those particles. You have still the same dispersion relationship, basically, so you get this absorption into the UV. That's a property that remains from the bulk properties of the semiconductor, but you get the excitonic peak. And if you have a smaller particle, you get a blue emission. A bigger particle, you get a red emission, so you have control over the band gap just by this quantum confinement effect. But what that means is you can use a single laser to excite several different size particles and get several different colors all at the same time. So here you see an experiment. Those are 5 test tubes. In each test tube you have different size quantum dots. You pass in a single laser beam of 488 nanometers, and you get all these 5 colors fluorescing at the same time. And what that means is you can go now into biology, and you can wavelength division multiplex biology. You can go into the living cell and paint different proteins. If you have the specific chemistry, you can go and paint different species, different proteins with different color particles. As added to this very basic advantage, you have other very good advantages. Those particles are extremely bright. They're much brighter than organic dyes. They're extremely stable. They don't photobleach. They have very narrow emission. That means you can pack many different colors along the optical axis. They have long fluorescence lifetime that allows you to do time-gated detection. They have different contrast mechanism. You can use them, for example, in a live cell with conventional fluorescence microscopy, and then you can fix a cell and go to the electron microscope, and you have high Z contrast from these metallic ions. So there are many, many good advantages. Company was started based on this concept, and you're all familiar with quantum dots. But what I want to do today is actually talk about disadvantages of quantum dots rather than the advantages. Obviously, working with quantum dots for almost 20 years now, we know 1 or 2 things about the disadvantages. One of the most striking disadvantages is what you see here in the movie. Quantum dots tend to blink. And there are hundreds, if not thousands, of papers relating to this blinking phenomena that was reported for the first time by the Bowendi group in 1996. There are many phenomenological explanations for this blinking, but there is no good molecular understanding of what this blinking is coming from, and we'll not spend too much time on this, but this is one of the big lemons of quantum dots. Obviously, if you have particles that blink, they don't give as much light as you would like to, because if they would not blink, they will be even brighter. And I'll give you another reason why blinking is not good. There are other major disadvantages. They are made out of toxic metallic ions, so they could be toxic for in vivo applications. It's very hard to control the surface chemistry and functionalize them in a way that you can target whatever you want to target. It's very hard to control stoichiometry, such that you have only one quantum dot binds to only one protein without any cross-linking. So there are many disadvantages, and over the last 2 decades, people have learned how to basically solve most of them. For example, blinking, there are some generations of quantum dots that almost don't blink at all, and basically there was a big effort in the field to make them non-blinking. My group has done quite some work on single particle tracking, and obviously for this type of application, quantum dots are ideal because they're very bright, so you can follow particle for a very long time. One of the most outstanding questions still in cell biology, unsolved to this day, very controversial, is the question of how membrane proteins are organized in the membrane. And maybe Tom will be able to answer that question. That relates to the question of lipid rafts. So our proteins in the membrane just undergo simple Brownian motion. They are dispersed everywhere and do simple, basically stochastic simple motion, or they are arranged in some small patches of different content lipids that actually are functional, such that when you want to signal in and out of the cell, you basically self-assemble several different proteins that phosphorylate and do things and so on. And if indeed those patches, or what people call in the field lipid raft, exist, what the size, what are the properties, what is the state of the lipids within those patches and so on. And there are many theories and many experiments that have lasted for the last almost 4 decades trying to answer this question. So our contribution to this field was to attach single quantum dots to GPI anchored proteins. Those are proteins that are post-translationally modified after they are synthesized by the ribosome. Basically, there is a covalent attachment of a lipid to this protein, and this protein is trafficked to the membrane. It crosses the membrane, the 2 leaflets of the membrane, and then it's anchored to the upper leaflet from the outside of the cell side into the lipid membrane. And doing a lot of biochemistry experiments over many years, people basically doing sucrose separation of different lipid content and see which type of proteins are in which band. It's well established that if lipid rafts do exist, they're supposed to be rich in cholesterol, for example, and sphingolipids, then those GPI anchored proteins are within lipid rafts. So our idea was to engineer a GPI anchored protein with basically an avidin facing the outside world, and come with a streptavidin coated quantum dot that will, and come with biotinylated quantum dot that will bind this streptavidin. In this case, we can target specifically only the protein of interest and watch that protein for a long time. So this is what the movie is. The red spots are those GPI anchored proteins, and the green patches are cholera toxin labeled with organic dye that bleaches very fast. So the movie is run several times over. You'll see at the beginning of the movie in a second. So here you see strong dye signal that bleaches very quickly. Quantum dots, these are single quantum dots, and you can see some of the proteins undergo large diffusion, quick diffusion, while others are kind of localized and hardly move. And after 6 years of taking thousands of such trajectories and really figuring it out, you can graduate and get a PhD and eventually a position. So the student who did that work is now a professor at USC. And that's the punchline. The work is published, and I'll give you the reference in a second. We very clearly see 2 population, bimodal distribution of diffusion constant basically attesting to the fact that the same protein basically feels two different environments within the cell membrane, which is a strong support for this lipid raft hypothesis. But think about the disadvantage of quantum dots. We talked about blinking. So what happens when you try to follow these trajectories and you have several different quantum dots that basically tell you where the proteins are, and they undergo diffusion, and those trajectories cross each other, and you get blinking at the same time. You obviously lose your data, you lose your trajectories. So this is another reason why blinking is not good. Okay, so this is the lemon, and now let's convert the lemon into a lemonade. So the work is really the work of a very talented postdoc, Thomas Derzinger, who is now back in Germany, actually trying to commercialize the work he had done. The idea comes from Jörg Enderlein, a very good friend and colleague from University of Göttingen, brilliant guy. And this work was aided by Ryan, Robert, Gopal, and Jamin. And now I would even like to turn off all the lights totally. Can we do that? Can I get help to do that? Just for one slide? There, perfect. Thank you. So if you look very carefully now, you will see the same blinking I showed you before, except that the conditions here are different. I've shown you a live cell movie, and I had very dilute labeling of quantum dots. Here we have a fixed cell, and we use antibody to target quantum dots to microtubule network, and those are very densely labeled with those quantum dots. But you still can see the same type of blinking phenomena as before. And what we can do with this blinking is basically we can go from here to there. So I will claim that what we do here, and I will go over the mathematical algorithm, we get here super resolution, we get here terrific contrast enhancement, and we get here three-dimensional sectioning, and all with paying very, very little. So the movie I've shown you can be described mathematically basically by this expression here. Every single quantum dot within this microtubule network has a brightness of epsilon, let's call it epsilon k, and it has a blinking function. This is random stochastic blinking, telegraph noise-like blinking of each of those quantum dots. We take each of those quantum dots and we deblur them to the far field using the Poinsett function. So that's the green function of the optical setup. And we add all the different quantum dots within that frame, with that field of view, to generate the movie where we get a signal where R represents a pixel within a frame and T represents a frame within a movie. Now, if we have fluctuations, the first thing that comes into mind, Tom, the correlation function. So we are going to do very simple second-order correlations function, but before we do the correlation function, we are going to subtract the mean. So we take the signal that the term that I just wrote on the previous slide, we calculate for each pixel the temporal average over the whole duration of the movie, subtract it from each of those pixels, and we get just the fluctuations over 0. And then we write the second order correlation function, and initially I'm going to write it in the most general case. We are going to assume that into one particular pixel within the camera, we are getting contribution from several different quantum dots. So let's assume we get contribution from two quantum dots, K and L. And so that's basically taking the term that we had before, plugging it for two different quantum dots, K and L, deblurring it to the far field, and this is the correlation function. Here we have the temporal correlation function between the two blinking functions of particle K and L, and here comes very strong assumptions for this method to work. I'm going to claim that individual quantum dots within this field of view are going to blink independent of each other. To what extent this assumption is correct or not, we can discuss later, but let's assume that this is the case. If that's the case, we have here basically a correlation function, or a temporal integral, or think about this as a dot product between two orthogonal wave functions, If you have two different totally uncorrelated telegraph noise functions and you do a correlation between the two, the correlation will go to 0. What that means is that the contribution to the signal from different quantum dots basically will go to 0. The only surviving term is if we basically look at the fluctuations of the quantum dots with itself over time. This is the only surviving term that is correlated, and therefore this term now is reduced to a simpler term here, which is basically the autocorrelation function of the fluctuation of quantum dot k with itself, brightness square, Poinsett function square. And that's basically the essence of the whole idea, because if you take a Gaussian and you look at the full width half max of that Gaussian and you square that Gaussian, the full width half max of the square Gaussian is basically square root of 2 narrower than the original Gaussian. What this term here tells you that by doing this algorithm, the Poinsett function of the image generated by this algorithm will be square root of 2 narrower than the original image. That's not much, but we'll talk how we can do better. Instead of relying on this mathematical term, this is basically a graphical description of what I just told you. So this is the field of view. These are the microtubule network or whatever. We have quantum dots. Let's assume that the distance between those two quantum dots is much shorter than the diffraction limit of light. We use large magnification such that each Poinsett function is going to be sampled by several pixels on the camera, let's say 3 by 3 to 5 by 5 pixels. That means that pixels correspond to about 50, 30 nanometer within the object. And we are going to take a movie as function of time, so each of those frames represent basically a frame within the movie. So you see here the response to the Poinsett function. This is a deblaring of each of those basically molecular level emitters that are deblared to the far field. Now think about the signal on this pixel here. The contribution to this pixel comes mostly from this quantum dot, and if you do a correlation function, this quantum dot blinks, you're going to get a relatively high amplitude for the correlation function. Think about the pixel on this side, same story. It comes mostly from this guy, and there will be a large correlation function. But pixels here are basically a superposition of signals coming from those two blinking objects. And as we add more objects, basically the correlation amplitude goes down. This is very similar to the situation in fluorescence correlation spectroscopy, FCS. If you increase the concentration of the sample to the point where you occupy within your confocal spot many, many different fluorophores, you lose the correlation. So that's basically the idea. So the algorithm is extremely simple. We take a movie. We take the value at each of those pixels as function of time. That creates a time trajectory, a fluctuating time trajectory. We take each of those time trajectories and we perform the second order correlation function. In fact, we can just suffice with the first term, time delay equals 0, and we get the amplitude of that correlation function. And then we generate a new image from this series of frames where the value of each pixel is the amplitude of the correlation function. That's it. If you remember something from this talk, that's what you need to remember. That's basically the whole SOPHY method, and basically the claim is that we are going to go from the diffraction limit picture to something that is better by square root of 2. So it actually works. Here is data, here is a cross section. Blue is far field diffraction limit, and red is SOPHY. So you see some filaments here in the microtubule network. But actually, you have much more than that. It's not only super resolution. Look at the original image here. You see a lot of blur here. That blur comes from two main sources, autofocus light and autofluorescence. Autofocus light scrambles between pixels, and autofluorescence is molecules at high concentration. Those two contributions are not correlated, and when you do the SOPHY analysis, they are basically correlated out. So the resulted image has terrific contrast enhancement, and as I will prove later, actually has sectioning properties as well. Now, this is a very simple algorithm that can be performed on the computer or on even faster on an FPGA or GPU. And you don't really need to go out there and buy a STED or Palm or Storm machines for a lot of money. If you have a fluorescence microscope and you take time-lapse imaging, as I described, you basically can do super resolution, provided that you have this blinking object. So here is an example of SOFI performed on a TIRF microscope, and this is an example of SOFI performed on a spinning disc confocal microscope. This is SOFI performed not on quantum dots. I started with quantum dots, but if you slow down the blinking of dyes, and all dyes do blink, but they blink on a very fast time scale. They blink basically intersystem crossing to the triplet state, and the triplet state lifetime will give you blinking effectively in the 10-100 microsecond time range. Mike Hanneman, Philippe Tinefeld, and Marco Sauer worked out buffers that contain electron donors and acceptors that actually can slow down this process, and you can slow down blinking of dyes down to several milliseconds, slow enough that the best CCD cameras, EM CCD cameras, actually can capture those blinkings. And here is SOFI of the same microtubule network, now labeled with dyes and not with quantum dots. So the method is really very simple. You need a fluorescence microscope, you need a good camera, and you need a sample that is labeled with blinking objects. Now, what is the real promise of this method compared to other methods? This is a latecomer to the super resolution field. You all are well familiar with Palm, Storm, D-Storm, STED, and so on. The idea here is that we can actually acquire the super resolution data very, very fast. I explained before that we need a movie to generate a single frame. And the question is how long that movie needs to be. It turns out it needs to be about 100 frames. So that means if I take a movie of thousands of frames, I can take a window of 100 frames and move that window along the movie and generate a SOFI movie. I can do this very fast, and here without much effort, we are getting super resolution imaging. This is a transient phenomenon. When we start the movie, basically we get super resolution within few hundreds of milliseconds. This is without making a big effort to be really very fast. We think we can do better than that. So SOFI at this point is not very high resolution. We'll talk about how we can make it higher resolution, but there is a trade-off basically between speed and resolution. You might know that Palm and Storm are terrific high resolution methods, but they require very, very long movies, and the reason is we are going to light up only very few fluorophores for each frame of the movie, use fancy fitting algorithm for each of those spots, but we want to make sure that each spot is isolated. That really slows down that process tremendously. While in SOFI, we can have half of the molecules or 3/4 of the molecules within field of view to be in the on state at a given time. We just need a little bit of blinking, and we can get this resolution enhancement. So can we go to higher resolution? Obviously, if you go to higher order correlation, you might be able to do this, and what you expect is that the Poincare function will be raised to n being the order of correlation. In fact, we are not talking about correlations here, but cumulants, which are superposition of different correlation terms. And indeed, this is an experiment that showed that point. We just spun some quantum dots on a cover slip, and it so happened that we had two quantum dots really close to each other. In the far field, we can distinguish the fact that we have two here. And we basically take a very, very long movie. In this case, I think we took 10,000 frames. Here is a cross-section, and what you see here in the cross-section, this is basically the raw data. This is a full width half-max diffraction limit PSF that you see here for one of these quantum dots, and you see background. Then since we have those movies, we can do correlation, and we can do the correlation at higher and higher order. So here is second order. You can already see the two emitters well resolved. And as we go to higher and higher order, the PSF shrinks more and more, and you can see this on the cross-section. So already at second order, the background is gone. It's uncorrelated background. So we get contrast enhancement, and then as we go to higher and higher order, we get narrower and narrower PSF, to the point where if you get to the order 25 in correlation order, we are at 50 nanometer. But obviously, we lose the most important advantage of SOFI if we do so, because this will take a long time. We need a long movie, and that is going to slow us down. So there is somewhere here a sweet spot where SOFI will provide speed with good enough resolution. So we don't promote the idea that we can do very high resolution. For this, you really need quantum dots that can stay stable for a long time. And there is something really interesting here on the physics side of things. Quantum dots blink in a power law, Levy flight fashion, not in a Poisson fashion. That means you get blink, there is no mean for this stochastic function, and you get blinking on all time scales. And in fact, as you go to higher and higher order correlation, you can see that the brightness of these two spots changes. That comes from this fact. Because quantum dot can turn on and stay on for minutes and then turn off, or can blink on a microsecond time scale. Imagine that you have one of the quantum dots very bright but doesn't blink at all. The other one is dim but blink fast. As we go to higher and higher order correlations, the guy that doesn't blink will disappear. The guy that is dim but blink a lot will get stronger. So this is the phenomena that you see here, and that can be corrected. In fact, Theo Lasse from Lausanne came up with an algorithm that correct for that effect. Now, I promised three-dimensional sectioning. Again, we spin quantum dots on the cover slip. Now we have a piezo on the objective, and we can change the height of the objective. So we can take movies at different heights. Let's say we have a step size of 50 nanometer, and we take movie every 50 nanometer in Z. We can look at the points of function. We basically we SOPHY the information for each of those sections, and then we render the information for all section in 3D. We can do this for the far field. You get this cigar-shaped PSF. And as we go to higher and higher order, you can see that the cigar is shrinking simultaneously in 3 dimensions along X, Y, and Z. And the reason is the algorithm doesn't really care if two blinking objects that are not correlated sit in the plane or along the optical axis. So we don't need the double helix for that. And we don't have a dynamic range of 1 micron here. In fact, we have almost unlimited dynamic range. It's not as good. We cannot locate in Z as accurate as a double helix can. But we have here a method that allows us to get information in 3D. Does it really work? So let's try to do this on cells. Same microtubule network, decorated with quantum dots, taking movie at different heights, taking this information into a 3D rendering, and if we put garbage in, we will get garbage out, because you don't expect the wide field microscope to give you any information along the z-axis. But if you take each of those movies at different sections and SOPHY it first, and only then 3D render it, you build the cheapest confocal microscope you can imagine. This is a piezo on the objective and blinking objects, and that's it. So now you can get 3-dimensional information. So here is just sections at different heights. And here is a cross-section along XZ, so blue is far field and red is super resolved information along Z. All right, so far I talked about performing autocorrelation function for the temporal signals of individual pixels. But I also told you that we tend to magnify quite a bit the image such that each PSF is sampled by several pixels. That means that fluctuation from the same quantum dot arrive at 2 adjacent pixels. Therefore, I am allowed to do cross-correlation between those pixels. So here is similar math as before, with few small differences. So again, I'm starting in a general case where I look at 2 different quantum dots, K and L, at 2 different pixels, adjacent pixels, R1 and R2. And when I work out this, first of all, only the 1 quantum dot case will survive, so K equals L. I get the same brightness square as before. I get this cross-correlation term, but I get here the same PSF squared, but I get here the argument for the PSF is now shifted by the geometrical average between the 2 pixels. And there is a prefactor that actually attenuates the cross-correlation with respect to the autocorrelation. What does that mean? If I have 2 adjacent physical pixels and I take the temporal signal on each one of them and I do the cross-correlation, I actually calculate what should be the signal in between those 2 pixels without the chip manufacturer providing a pixel there. Okay? This is not basically interpolation between the two signals here. This is actually weighing, based on the amount of blinking I get here and here, I find what the signal should be here. So this is one cross correlation. I can do this diagonal cross correlation, and I can do this cross correlation, and now I generated new pixels that were not there. That allows me to increase the number of pixels on my camera by a factor of 4. I can do this even more. I can take higher order correlation. For example, I take 1 part here and 3 parts here. That will generate this pixel, 2 and 2 this pixel, 3 and 1 this pixel, and now I have 16 times more pixel information as before. So if I don't change the magnification and I want to zoom in and out, I have still the ability to keep enough pixels there to get the super resolution image. Okay, so now let's try to put everything together. So what Thomas did, he generated a particular pattern where for each of those spots he generated, in this case, just a simple perfect Poisson blinking function. They're all not correlated. Deblur all these spots to the far field using a pointed function, and then try different algorithms. So here is 4th order autocorrelation. Now, I also mentioned that when we do the cross correlation term, in contrast to the autocorrelation term, there is this prefactor that attenuates a signal. So if I plot on the simulated data all the autocorrelation functions, basically from individual pixels, and all the only nearest neighbor cross correlation between all pixels, including the diagonals, I get this pattern here where the darker pixels are the cross correlation terms and the brighter pixels are the autocorrelation terms. Now, I can ask the computer to actually correct for this and basically smooth that surface, and that accounts to basically measuring this function. So basically, by computational method here, I can measure the pointed function without measuring it. And if I have access to the pointed function, I can use it for deconvolution. So now if I go to order 4, for example, and I use deconvolution on top of this, because now I have handle on the pointed function, I basically get very high resolution picture. So in this discussion, I started with a square root of 2 enhancement. I told you if we go to higher order, the resolution enhancement scales like square root of n. But if I do this cross correlation trick, I now can basically go and scale like n. So if we go to order 4, we start with a PSF which is 250 to 300 nanometers. We are now 65, 70 nanometer resolution enhancement. Order 4 is very reasonable, and that's what we believe will be the sweet spot. So the method works, and now you can try to use it for different colors. So here you see a work where we colocalize P-body proteins. P-body are small organelles that are self-assembled to store and process messenger RNA. And there are some reports that they are associated with the microtubule. So we label microtubule with one color quantum dots, and we label the P-body proteins with another color quantum dots, and basically we do colocalization, conventional immunocytochemistry colocalization, but now at super resolution. We also found some interesting, intriguing data about fully assembled P-bodies that look like donut shape, but that needs some more work to be proven. We can do 3 colors. But the big promise, as I told you, this method really allows us to speed up super resolution. We compromise resolution, but we can go quite fast. And for that, we would really like to use the method, and that's where we think the big payoff is to look at cellular processes which are quite dynamic. For example, polymerization of tubulin and actin polymers as function of all kinds of cues that arrive at the cell. What we have done here, we are using microinjection of quantum dots that are functionalized with monomers of tubulin, and those are incorporated into the growing tubulin network within the live cell, and you can see the blinking. So the vision is really to take this type of technology to introduce blinking into live cell and use this method to look at fast processes at better resolution than the diffraction limit. Obviously, quantum dots are not the right way of introducing blinking into cells. but we can talk about this later during the questions session. Just a quick word on how we did this. We basically took full polymerized tubulin and then labeled the whole tubulin fibers, and then basically depolymerized the product to make our reagents, and we injected those reagents using a new method of microinjection developed by colleagues at UCLA called the nanoblade. This is, I don't have time to go into this, but this is light-assisted microinjection without actually puncturing the cell membrane with your pipette. And that allows very efficient delivery of quantum dots. Another approach of introducing blinking is binding unbinding. This is the work of Marcel Bouchez and Alan Wagoner from Carnegie Mellon. They call this technology FAP, fluorogen activating proteins. They developed a single chain fragment antibody that binds malachite green. Malachite green is a normally quenched dye that is in the dark state, but when it binds to its antibody, it turns on. The affinity of that dye to the antibody is not that high. It binds and unbinds. So if you are basically expressing a protein in the cell that has this antibody fused to it, and you immerse the cell within those dyes, the dyes are permeable, they will go in, bind, unbind, and you can get blinking not by photophysics, but blinking by binding, unbinding. That's the vision for this type of technology. It's not working that well, needs much more work. But obviously the most promising approach would be to get fluorescent proteins to blink. And it turns out that DROMPA does that to some extent. What you see here are focal adhesions labeled with DROMPA. And we worked out some conditions using LED illumination. You need actually very low light level illumination, single color, taking movie, and now our microscope and super resolution method consists of protein cloning, fusion, and a simple fluorescence microscope with LED or possibly even a lamp illumination. That's what it takes. So we think that this type of approach really is the right approach, where the entry level for biologists will be very low. It will be very simple to do. So that concludes the part on SOFI. Our last word, we are not the only ones doing SOFI nowadays. There are many groups following up, and this is the work of Jean Zhang from Johns Hopkins, where they did exactly that. This is DROMPA labeled mitochondria that they SOFI'd and got very nice data on. And the method I described doesn't really rely on fluorescence. You can use it for any imaging modality where the objects contain elements that basically blink. It can work with absorption, it can work with scattering. So here's an example of plasmonic particles that are used for SERS, for stimulated emission Raman spectroscopy. You basically have a dimer of silver particles, and you put organic dyes within the polymer here, and those blink, so therefore you can SOFI them as well. So this is now scattering result, where you can see here super resolution enhancement for scattering, not for emission. All right, skip that. Again, all the credit goes to Thomas and to Jörg and the people who helped. And if you allow me, for the last few minutes, I'm going to talk about a newer project in the lab. This is, again, related to quantum dots and quantum confinement. The work is the work of a very capable EE student, Philippe Park, helped with many other people. And I'll go very quickly through the issue here. This is a neuron, and neurons communicate within themselves within the brain by electrical currents called action potentials. This is action potential measured by the patch clamp. The patch clamp technology got Naren Sackman the Nobel Prize. It's a 4 decades technology. Terrific technology. We have some of the psychiatric drugs are basically really based on the invention of this methodology, but it's invasive. It's not useful for imaging. Cannot perform multi-spot detection, long-term measurements, in vivo recording, and so on and so forth. And obviously, you can multiplex this idea, and this is called the Utah array. And the last issue, the March issue of Nature Nanotechnology, had 3 back-to-back papers by 3 groups, all going to this type of patch clamp array electrode that will allow you to measure electronic signals inside the brain from many, many different locations and basically build an image from those electrical signals. So this is the work of Charlie Lieber using basically nano electrodes that are actually individual transistors. This is a work of Hong Kong Park, and this is the work of Chu from Stanford, all basically going after those miniature nanotechnology-based electronic measurement of those electronic signals. But there is another field that many people have tried to contribute to for many decades, which is voltage sensing. If we can have fluorescence-based detection of voltages on axons, that will be so much easier. High throughput multiplex detection of electrical signals. So the idea would be that you launch an electrical signal and basically you follow some change in property of your dye. That could be different color, that could be different polarization, that could be different lifetime. And many people have contributed to this idea of voltage sensing dyes, but the truth of the matter is that after intense 4 decades, we still don't have good enough voltage sensing dyes. Roger Chien developed his 3rd generation. By the way, his highest cited paper is not about GFP. His highest cited paper is about voltage sensing. So he has been at it for at least 3 decades. And he just published another voltage sensing dye, which is very intriguing. And in that paper, he summarizes 3 different technologies that he developed over the years. So the first voltage sensing dye is electrochromic, basically working on the Stark effect. The second works on FRET, where you have the donor on one side of the membrane and the acceptor is free to move within the membrane. Depending on the voltage, you have this majority moving in and out. This turns out to be slow and capacitive loading to the membrane. And this most recent dye has a quencher that the amount of quenching depends on electron transfer, and electron transfer depends on the voltage on the membrane. So this is a much better voltage sensor, and I think it will be very, very useful. Adam Cohen from Harvard just published 2 papers, phenomenal, where he actually took bacterial rhodopsin and managed to make it a voltage sensors. So this is basically a genetic tag that can measure voltage that will work really well together with optogenetic tools that Karl Deiserhoff developed for the bacterial channels that you can modulate with light. So there is a lot of development in this field, and I think that especially this type of technology will be extremely useful for neurobiology. So what we do is not that. It's going to be useful not for cases where you can transfect or make transgenic animals. This technology is really good for cells, tissue culture, small animals, and so on, but it's not good when you want to work with a wild type. So we would like to have a nanotechnology tool that can do the same. It's not a genetic tool. And we go back to this quantum confinement business that I described before. So David Miller and Daniel Shemla worked out the quantum confined Stark effect in the early 80s in Bell Labs. I joined the Shemla group and worked in these topics. So for me, it's really a very interesting closing of a circle, coming back to this type of ideas after 25 years. And the idea here is very simple. The same exciton, particle in a box. We put this box under voltage, and the box tilts, energy levels basically lower, and the transition now from the bottom of the conduction band to the top of the valence band is redshifted compared to the unperturbed well. And that has been measured in quantum wells very nicely. So you can see here the banded shift as function of voltage putting on this well. So what we wanted to ask is, can we measure this action potential? And I'm not going to provide an answer because we are not there yet. I'll just show you work in progress. Can we measure this action potential with the same type of material like as quantum dots? This is a nanotechnology-based small particle. Can we do it at room temperature? Can we do it inside the membrane? And can we do it on the single molecule level? Mind you, none of the voltage sensing dyes that exist today are single molecule voltage sensing dyes. So, what is the action potential? The action potential is 120 millivolts over 4 nanometer of membrane that translate to 300 kilovolts per centimeter. Most materials break under this voltage. It's quite amazing how the membrane of a cell can withstand this type of voltage. Okay, so what we did, we synthesized all kinds of nanomaterials based on these colloidal chemistry methods. This is a quadium selenide nanorod. This is a seeded nanorod where we start the synthesis of the rod with a quantum dot. So we have a quadium selenide dot that we synthesize a rod on top of it. This is type 2 quantum dots where the minimum for electrons reside in a shell and the minimum for the holes reside in the core of this material. This is a asymmetric heterostructure where we have basically a nanorod where one type has this band alignment and one type is this band alignment. Type 2 means that the minimum for the electron and the holes reside in different materials. So it is a very good candidate for charge separation. And even a more complicated scheme where we have both type 1 and type 2 transitions. Here is a table of all the materials we look at, high resolution TEM pictures, band alignments and so on. I'll try to move quickly because I'm running out of time. We build a dedicated single molecule setup to do that. So we have a cover slip here on top of the objective where I have patterned electrodes by lithography and we spin coat our nanoparticles in between the gaps between those electrodes. We have high voltage that we can apply on those electrodes. Camera is synchronized with the pulse that activate the high voltage. And we have here basically the detection arm. There is a slit. You see an image of several different nanorods here, the bright spots. We then close the slit just to limit the number of particles we look at. Then we add this prism and we disperse to get the spectrum. And basically now we run the experiment as a movie. We turn on and off the high voltage, and the video acquisition is synchronized with those on and off pulses. So the raw data looks like this. You have this checkerboard. White represent high voltage, black represent zero voltage, and the data looks like this. So here is a movie. You can see here the spectrum at 0 voltage, and then at high voltage the spectrum is dimmer and shifts downwards. So we get this zigzag. The way we process this data, we basically either integrate along the wavelengths the total intensity. That's what is plotted here in blue. You can obviously see the blinking period. This is the same famous blinking from the Sophie story. But you also see during the on time, you see this zigzag where the spectrum is shifting. This is just the intensity getting dimmer or brighter. We can also fit each of those spectrum and look at the peak position of the spectrum. This is shown here in green, and that basically shows the spectral shift as function of voltage. You see very nicely the zigzag due to this Stark effect. We then take basically all the on-state spectrum and average it to one spectrum, all the zero voltage spectrum and average to another spectrum, and this is basically just to beat the noise. So what you see here for this simple nanorod from cadmium selenide, the blue is the zero voltage spectrum, the red is the high voltage spectrum. You see a slight shift and a slight decrease in intensity. For seeded nanorod of type 1 or quasi-type 1, signal is much better. You see a bigger decrease in intensity and a bigger shift. And the best sample gives us a really pretty nice specular shift. This is stark effect at room temperature on a single molecule level, where we get 13, 14, 15 nanometer shifts. So we have done some attempts to orient those nanorods along the field lines within the electrodes by dielectrophoresis, but those attempts are not very good. So therefore, we get all kinds of shifts during the experiment. Here in a histogram showing many shifts for many quantum dots. This is sample 2, that is type 1. We don't get much stuff, but the outlier for sample 8 really go to pretty large shifts. Those are the nanorods that are lined along the field lines of the electrodes. So we have pretty good voltage sensors on the nanoscale. We can do more than that. We can actually change the voltage. So when we have this square pulse, 0 volt high voltage, we can go 0 to 50, 0 to 100, 0 to 150, and look at the voltage dependence of the shift. And for different samples, we get different voltage dependence. So if we separate for sample 2, for example, we get this parabolic behavior, and this is what you expect for classical Stark effect. But for sample 7, which is an asymmetric nanorod of type 2, we actually get more or less linear behavior of this voltage sensing, which is really very nice because you can measure the polarization, the orientation of the field. And the explanation for this is very simple because we have this asymmetric structure. Depending on the orientation of the external field, when we have a type 2, we excite an exciton. Electron goes to one material, hole goes to the other material. In one field configuration, the wave functions are going to be separated even further. We are going to get even more redshift. In the opposite field orientation, the wave functions are going to be pushed against the interface between the two materials. And that gives rise to this linear behavior. We can actually reproduce this by performing self-consistent calculation, Poisson-Schrödinger coupled equation. I won't go through that. And we can actually, by simulation, get the same type of behavior. In fact, we can fit even the data and find the orientation of the nanorod. That's the only free parameter. And we can extract from the calculation the Coulomb interaction strength. And obviously, for type 2 materials that separate the electron and hole pair, we basically get lower Coulomb interaction, and those are better voltage sensors. So to conclude, these are the better sensors, and they have really nice properties compared to existing voltage sensing dyes. I won't go through that. And obviously, the main question is how to get them into the membrane. So this is the membrane, and we want to put a large particle, inorganic particle, into the membrane in the right orientation. We have not done so as of yet, but we are working on this. We thought about 2 approaches, or 3 approaches actually. One would be to put different surfactants on the surface of this nanorod. And there is some evidence that one might be able to do this because you have different surface energies on the bases of the cylinder compared to the circumference. So we can put different type of surfactants, hydrophobic here, hydrophilic here, such those particles self-assemble into the membrane. But what we chose to do first is to try to put alpha helix transmembranes, peptides, on the surface of quantum dots. We have quite experience with peptide coating of quantum dots. We have done a lot of work in this area. Now we modified this approach by putting these alpha helix transmembranes, and we do have evidence, but doesn't tell us that those particles go inside the membrane. They just tell us that they are associated with the membrane. So here you see giant vesicles decorated with those peptide-coated alpha helix voltage sensors. Here is a confocal sequence, different sections, and then we can take those vesicles and deliver them to the cells, and they will fuse into the cell, so we can deliver those quantum dots to the cell. But still, there is a lot of work that needs to be done such that we can put them in the right orientation in the membrane. The work is the work of Philippe, and thanks to many people, current and former in the lab, and many collaborators, and thank you for your attention. Yes?
For a single quantum dot that has a 20 nanosecond lifetime, what's the spectral bandwidth of the emission? 25 nanometers, or is a single dot emit a much narrower color?
Oh, of course, it will emit much narrower. Of course, you have spectral diffusion on a fast time scale that you integrate over.
And so a single dot will spectrally diffuse over 25
Right.
nanometers?
Yeah.
Or all the ensemble of dots, how's that?
No, the 25 nanometer is the spectrum of a single dot at room temperature.
You mentioned that you assume the quantum dots were uncorrelated.
Right. So obviously, at some distance, when they are close enough, they will start to talk to each other. They will start to fret, and fret will synchronize those blinking behaviors. So, in fact, we can see some of that. Philippe did observe when he goes to very high concentration, he can see some correlation. But we never really looked into this very carefully. There is another paper that I mentioned to Tony before by another group. I think it will be really interesting to study blinking as function of distance between quantum dots. But, you know, the fact that SOFI works at those labeling densities, they are more or less uncorrelated. But that definitely be a limitation when you go to very high dense labeling.
In your correlation functions, you have time delay between.
What?
You never mention it in your results.
No, the time
Is
that
that zero or
the correlation function, it's an autocorrelation function of the pixel with itself at different times. So that basically tells you to what extent the blinking is correlated until it goes away.
when you generate your pictures, what time delay you are using?
Zero.
So
Zero. It's just a DC
It's
field.
just decoration for the.
It's just the amplitude of the correlation function at time equals zero. Well, that is not entirely correct. That doesn't work well for the autocorrelation. That works really well for the cross correlation of adjacent pixels. Because for the autocorrelation, you'll get a short noise in the zero term. So you want to go 1 tau away for autocorrelation, but you can use tau equals 0 for the cross correlation.
So there's no additional correlation.
Well, but you can do SOFI in many different ways. There are infinite ways of how to do SOFI. You can integrate the correlation function and stop it at some point. You can do all kinds of different things. We did the simplest thing, which is take the first term of the correlation term.
Do you try to optimize?
No. There is a lot of work that needs to be done. There should be a theory that talks about signal to noise, signal to background, what is the ultimate resolution, all kinds of different things like that. I'm actually looking for help. There is a lot of work that could be done based on this concept. Yeah.
So you mentioned that the autocorrelation SOFI algorithm works really well behind an autoplane. It seems that if you have two quantum dots that are located, like one behind the other, like small distance, you get no signal.
Well, I was cheating, and the reason I was cheating is the way the data was taken here was at different times. Okay? In order to do it properly, you need to take the different Z sections at the same time. Right? Because you want to look at correlated fluctuations. And in fact, you can do that. Ober and Phil just published a paper where they have 4 different foci measured by 4 different cameras simultaneously. My former postdoc, who is currently visiting Genelia Farm, Maxime Dahan, is using diffractive optics, a smart hologram that basically takes the image and generates 9 different foci on the camera. That's another approach. So there will be approaches, I think, that will allow you to take images from different foci simultaneously, and then you can do temporal correlation among Z, which I haven't done here. What I've shown here was each section taken at different time. So it's sequential.
Okay, I think we're out of time.